SB015 Biology Practical Test to Lab Report. Hi, this video belongs to me, Audi. Let's dive into the world of life science by learning DNA isolation. The objective of this experiment is to isolate DNA from plant tissues. The theory of this experiment is each chromosome is a single thread-like structure made up of long molecules of DNA combined with histone protein. The DNA molecule is made up of small sections called genes. Before cell division occurs, DNA replication occurs forming two chromatids. In these chromatids, there are identical genes called alleles. In this experiment, we will rupture fruit cells, thus releasing their contents such as protein, DNA, RNA, lipids, and various small molecules. The DNA is suspended by alcohol as a supernatant layer. Moving on, materials and apparatus. For materials, we use soft fruits like banana, tomato, onion, and kiwi. Then we use ice chill alcohol, ice cubes, and salt detergent solution. For apparatus, we use mortar and pestle, beakers, a tube rack, a strainer, boiling tubes, and measuring cylinders. Next, the procedures. Step 1. Pour 10 ml of ice chill alcohol into a boiling tube and place it into a beaker containing ice cubes. Step 2. Slice and mash the fruits using a mortar into fine texture, like so. Step 3. Transfer the mashed fruit into a beaker. Step 4. Add 100 ml of the salt detergent solution. Incubate the mixture in a water bath at 60 degrees Celsius for 15 minutes. Step 5. After 15 minutes, sieve the mixture with a strainer and collect the liquid in a clean beaker. Step 6. Fill in half of the boiling tube with sieved liquid. Step 7. Very carefully pour 10 ml of ice chill alcohol into the side of the boiling tube at a flat angle. Make sure both liquids do not mix and alcohol form a separate layer on the top of the sieved liquid. And lastly, Step 8. Observe and draw the extracted DNA between alcohol and the sieved liquid in the clear container. Next, result and observation. As you can see, this is what we got after the experiment. Now, let's observe what can we see in the boiling tube. In the test tube, the yellowish bottom layer is the sieved liquid obtained from the mashed fruit mixed with the salt detergent solution. The top clear layer is the ice chill alcohol. Alcohol is less dense than the sieved liquid, causing it to float. The white clumpy substance is the extracted DNA. The crude DNA appears cloudy and has a texture which is stringy, fibrous, sticky, similar to mucus. And lastly, discussion and conclusion. Let's discuss on these questions. Question 1. Why is this step taken? Mechanical destruction using mortar and pestle. Firstly, to break open the cell. By applying force, the pestle grinds and crushes the plant tissue into smaller fragments. This releases the DNA in the nucleus and plasmid. The plant cell walls need to be broken down effectively to access the DNA for the experiment. Question 2. Why is this step taken? The addition of salt detergent solution. The positively charged sodium ions from the salt protect this negatively charged phosphate group in the DNA backbone. This promotes clumping for easier separation, while the detergent disrupts the lipid of the cell membrane, releasing DNA from the cells. Question 3. Why is this step taken? The addition of ice-chilled alcohol. Alcohol must be chilled before the experiment. This can help in DNA precipitation. DNA is soluble in water, but it is insoluble in alcohol. This can help in formation of visible precipitation. The crude DNA will appear as a cloudy, white, streamy substance. Question 4. Why is this step taken? Leave in water bath at 60 degrees Celsius. The heat from the water bath denatures proteins that are attached to the DNA. This also accelerates the cell lysis process. The heat from the water bath also enhances the action of the detergent. This will help to break down the cell membrane and nuclear membrane more effectively. Last question. What are the precaution steps taken during the experiment? First step, the alcohol should be chilled before the experiment. Second step, avoid formation of soap bubble when stirring the salt detergent solution. Third step, pour the alcohol very carefully into the side of the boring tube at a flat angle to ensure both liquids do not mix, like so. Before we end, what is the conclusion that could be made? The conclusion is, the plant DNA was successfully extracted and visible as a precipitate. The detergent effectively lyses the cell membrane and nuclear envelope. The salt helped protecting the charges, enabling DNA to clump together. The water bath aided in protein denaturation, enhancing cell lysis and protein denaturation. Lastly, the addition of ice-chilled alcohol precipitates the DNA. Therefore, the objective of the experiment has been achieved. Yay! That's all from me. Thank you for listening. Biology A. Bye-bye.